Welcome students, I am Pooja Sakalkar, Assistant Professor at Kamal College of Arts, Science and Commerce, Nuve. The course title of today's module is Biodiversity 1, Microbes, Algae, Fungi and Bryophytes. The title of this module is Lichens General Account. To give you an outline of this module, we will begin with a brief introduction, then we will look at the details about the photobiont and the mycobiont. We will look at the occurrence of lichens, its growth rate and finally the classification. This module defines a lichen and describes its characteristic features. It explains the features of photobiont and mycobiont. It outlines its classification and it outlines grouping of lichens based on other characteristic features. To begin with, introduction. Lichen is an association of a fungus and a photosynthetic symbiont resulting in a stable thallus of specific structure. Lichens may also be defined as a stable self-supporting association of a mycobiont and a photobiont. So, we can say that lichen is an association between a photobiont and a mycobiont, wherein the photobiont can be a green alga or a cyanobacterium, whereas the mycobiont is a fungus. Now, the characteristic of the lichens are unique and neither of the partners alone have those features. Details of photobiont. The photobiont, that is the alga or the cyanobacterium, carries out photosynthesis. Most of the photosynthate produced, that is about 70 to 80% of the photosynthate produced is incorporated into the fungal hyphae. There are around 37 genera of algae and cyanobacteria known to form lichens. Common chlorophyta genera are Treboxia, Chlorella and Cephalurus, whereas the cyanobacterial genera are Nostoc, Anabina, Calothrix and Cytonema. Now, let us look at the details of the mycobiont, that is the fungal partner. The fungal partner provides the algal cells with water and nutrients. It also protects the algal cells from high light intensity. There are approximately 14,000 species of lichenized fungi known. Most of these are Ascomycota members belonging to Discomycetes or Pyranomycetes. Some of them may belong to Loculoascomycetes. Some of the Basidiomycota members are belonging to genera Dictyonema, Omphalina and Multiclavula. But compared to the Basidiomycota, most of the lichens have Ascomycota as their fungal symbiont. Now, let us discuss about the occurrence of lichens. Lichens have a wide distribution worldwide. They grow on soil, rocks, trees and even walls. They grow in a variety of habitats from cold to hot, from arid to moist conditions. A number of species occur only on seashores. It forms the dominant vegetation in the polar regions. Lichens are known to be the pioneer species in primary succession, as it involves in weathering of rocks and soil formation. It is important to note here that the autotrophs, that is the alga or cyanobacterium, may be found free living, but the fungal symbiont is never found free living. The growth rate of lichens. Lichens exhibit a very slow growth rate. Most of the lichens grow at the rate of 0.5 to 8 millimeters per year. Their growth rate is affected by factors such as light, temperature and moisture. Now, let us discuss about the classification of lichens. Based on their fungal partner, lichens are classified into two: ascolichens and basidiolichens. Ascolichens are those lichens whose fungal symbiont is an ascomycota member, and basidiolichens are those whose fungal symbiont is a basidiomycota member. Now, let us look at other groupings of lichens. Lichens may be grouped based on their thallus structure into 5 types, that is leprose, crustose, foliose, fruticose and filamentous. Leprose is the simplest type of thallus. Fungal hyphae envelop small clusters of algal cells and grow superficially over the substratum. They have a powdery appearance. Next is a crustose lichen, wherein the thallus is closely adhered to the substratum and has crust like appearance. The photobiont cells are covered by distinct layer of fungal tissue. The next type is the foliose lichen, wherein the thallus is flat, leaf-like, lobed and attached to the substratum with the help of rhizoids. The fourth type in this group is the fruticose lichen, wherein the thallus is well branched and erect or pendulous. And the last group is the filamentous lichen, wherein the thallus is a mass of thin stringy filaments made up of the photobiont, which is surrounded by the sheaths of fungal filaments. So, these are the 5 groups based on the thallus structure of lichens. In this image you can see this is the leprose lichen, this is a crustose lichen which is adhered to the substratum, this is foliose lichen which is lobed and leaf-like, this is fruticose lichen which is erect or pendulous and little more complex, and this is the filamentous lichen. The next grouping of lichen is based on the habitat in which it is found. So, based on the habitat, lichens can be grouped into 6 groups. First is the corticolous lichens which grow on barks of trees. Second are the lignicolous lichens which grow directly on wood. Third is the saxicolous lichens which grow on rocky substrata. Fourth are the terricolous lichens which grow on soil. Fifth are the marine lichens which grow on siliceous rocky seashores, whereas sixth are the lichens that grow on hard siliceous rocks in fresh water. And finally, the grouping of lichens based on distribution of algal component in the thallus. So, depending upon the distribution of algal component in the thallus, lichens may be grouped into homoisomerous or heteroisomerous groups. Homoisomerous group is a group where the algal component is uniformly distributed between the fungal components. The thallus here is simple in composition and is not much differentiated. As you can see in this image, the algal component is distributed throughout the fungal component. Now, the second group here is the heteroisomerous form, wherein the algal component is limited to a certain area. The thallus thus has a layered structure and is much differentiated. So, as you can see in this image, the algal component is restricted to this algal zone or the algal layer, and the rest of the thallus is the fungal component, which is much differentiated into the upper cortex, the lower cortex, the medulla. These are the references for this module and these are the web links. Thank you.